All right, today we're talking about one of those proteins that every biochemistry and biology class covers, and every test expects you to know about this one. But it is for a good reason, and this is hemoglobin. We're going to talk about hemoglobin for this entire video, and you'll see how important it is. And you already know this, right? You already know this. You've taken lower level classes where they talk to you about how hemoglobin transports our blood. But what does that mean? What is blood made of? Because this is biochemistry. We're not just going to look at the colors and those cartoons. We're going to look at the actual proteins and the actual chemistry and things like that. And by the end of this video, you should really know about how hemoglobin, the red blood cells, work from our lungs to the tissues and back. And it's a great story, and it is a great example of how proteins work. So I have it here as hemoglobin and myoglobin. It's a model in allosteric regulation and multi-complex cooperativity, right? So we talked about these when we were talking about enzyme regulation and allosteric regulation and things like that. But I don't want you to be confused here, right? Hemoglobin can be considered an enzyme, but it's mainly a transport protein, right? It's its own class, right? It's a transport protein transporting oxygen from the tissues and things like that. But it is a few things happen to the chemicals that bind to it, right? And we'll talk about some of these things. And whenever you have a protein and then something binds to it and transforms, what's that? It's an enzyme, right? It catalyzes reactions, but that is not the main role. The main role of hemoglobin is a transport protein. So I want you to kind of know those distinctions and not be confused. But even though it's not a canonical enzyme, it is a good model to study allosteric regulation because it can be allosterically regulated by many factors that we'll talk about, but also because it contains multiple subunits that cooperate together, and we'll talk about that. But going back a little bit, you have learned about your circulatory system, right? In biology, you learn about how your blood circulates and things like that. And it's really good. This is how we literally stay alive and breathe and all that stuff. So I love this graph, but it doesn't tell the whole story, right? So it tells you that yes, blood circulates and then bad blood goes back, veins and arteries and stuff like that. And the heart is the heart is at the heart of this. But yes, it is really at the heart of this, and it just pumps all this blood and facilitates it. But the transport is made by these red blood cells, right? So this is a good overview. But then when you zoom in, you'll see that on your arteries and things like that, in your arteries, you're gonna have the red blood cells, right? They are red. And then because they are red and they are there, then what we're gonna ask you is like, okay, these red blood cells, what's in there? What's making them red? Okay, let's zoom in more. If you zoom in more, you'll see that these red blood cells have a lot of what? A lot of proteins. And one of the main proteins in these cells, this is kind of weird, red blood cells, but we don't have enough time to talk about that. But they have a lot of hemoglobin, right? Hemoglobin. And why do they have a lot of hemoglobin? Because hemoglobin is going to carry oxygen. And one of the things that makes it carry oxygen is what? It's iron. And because of iron, that is why we have red, because iron is red. We have a bunch of irons really you have 4 irons on a hemoglobin um, complex, right? Because you have 4 proteins per complex, right? So you have 4 irons, but they are red. Then you have a lot of these into the red blood cells, and guess what? It makes it red. And then it makes your blood red, right? So this is why your blood is red. It's because of iron, and we'll talk a little bit more about that. But in this chapter, we will even zoom in more and look at what is happening at the protein interface, right? What is happening to that iron at the bottom, right? What about the heme group? What about the protein? What is the exact interfaces? Because again, this is how we are literally kept alive. And these interactions are important. And the beauty of this is that we can understand this, right? The beauty and the superpower of biochemistry that I really like is that you can understand these types of process, because after you understand protein structure, protein binding, you understand the amino acids, the pockets, the chemistry, and all that stuff, then you can explain all these processes at the chemical level, not just superficial biological memorization. You can actually understand and explain what's happening. So one of the favorite questions that I like to ask students is, hey, tell me what happens to oxygen from the moment you breathe it to the moment it gets to where it is supposed to go. And where is it supposed to go, by the way? We will get to it, but the final destination of the oxygen is actually in the mitochondrial membrane, which is pretty cool. But it's in the mitochondrial membrane in the tissues. So one of my favorite questions is telling me the journey, not just like they take a ride on the red blood cells, but you can tell me exactly how they bind and where they bind on all the changes. And by the end of this video, you should be able to easily tell me that journey, no problem. All right, so another kind of, I got these images from the internet, but another one that I liked pretty well is you have a red blood cells or erythrocyte, and if you zoom in, you see hemoglobin, and then if you zoom in into the hemoglobin, you don't see just this. I don't like this one. I don't like this one. I've changed my mind. You do not see just this when you zoom in. Yes, when you zoom in into the quote unquote active sites, you will see a heme group, but you will also see an iron and a protein, right? So it's not just a heme group. But let's go back. Let's go back and kind of look at the biochemistry of it, right? So these proteins, and we will talk about hemoglobin, but also we will talk about myoglobin as well, right? So we call these heme binding proteins. They bind to the heme group, right? They bind to the heme group. And then this is again going to talk about the quaternary structure of the protein, which is the complex formation, right? This is a tetramer, right? This is a tetramer. Remember, you can have a dimer, trimer, tetramer, and so on and so forth. So hemoglobin is a tetramer, but guess what? Myoglobin is it's a monomer. The myoglobin is a monomer, doesn't have complexes, and we'll talk about that and the differences, right? So that's the reason why we study them, because they are really good models to explain all these concepts, but also they are very important in our bodies, and we definitely need to know all of these details, right? So, and also another reason too is one of the most studied proteins, right? Some of the proteins, the first structure, the first crystal protein structure ever solved was of hemoglobin, right? So, this is a classic example. But what we're going to look at is, yes, they transport oxygen, right? They transport oxygen. You have myoglobin and hemoglobin, right? And if you remember correctly, we talked about how cooperative binding increases increases the protein activity, right? Increases protein activity, but it increases it exponentially, meaning that we have cooperativity. So if you have this thing that has, this protein that has a certain activity, if you add another protein that has a similar activity, it will not be like times two, it will be times more, because they have cooperative binding. The binding of the substrate in one subunit influences the better binding in the other one, right? So that's what we call cooperative binding, right? But another thing that I want you to be able to tell me, and this is another one of my favorite questions, is for you to tell me the differences between hemoglobin and myoglobin. What are the differences between hemoglobin and myoglobin? And by the end of this chapter, you should be able to tell me about 5 to 10 differences, right? Well, what's the first difference? The first difference is myoglobin is a monomer, hemoglobin is a tetramer, right? One has one subunit, the other one has four, right? So that means that one is very small, one is pretty big, which is okay, right? And then this is very important, right? Myoglobin, right? Not myoglobin, but hemoglobin. Not only is it a tetramer, it's what we call a heterotetramer. Heterotetramer. Why? Because you have hetero and homo, right? So if you have like a homotetramer, all of the four subunits are identical. But a heterotetramer, all of the four subunits are not identical, right? So for hemoglobin, you have two that are identical and then two that are not identical. Two that are identical and then two that are identical. And we'll talk more about those, but you have alpha chains and beta chains, right? So two differences so far, two differences between hemoglobin and myoglobin. The third difference is going to be a kinetic curve, right? If you look at a kinetic curve, and this, by the way, is something that we will come back to a lot. So I really want you to understand this, right? I really want you to understand this because this is a kinetic curve. And if you remember, when we were talking about enzyme and enzyme activity is very hard to measure enzyme activity because different enzymes do different things. So we do not have a universal way to measure enzyme activity. You look at that enzyme or that protein, and then you're like, oh, okay, what is the product? What is the reactant? What can I measure? Can I measure the disappearance of the reactant? Can I measure the appearance of the product? So each enzyme, you tailor it. You tailor measuring the activity on the kind of assay that you're going to do. You tailor it to that enzyme activity and the signals that it's giving. So for hemoglobin and myoglobin, right, we use oxygen. Oxygen is a gas, it's easy to measure, right? So I want you to understand this because again, it's not just my class. Other people in the future where you go will expect you to know this and be able to interpret these type of curves. It is an oxygen binding curve, right? Or we also call it oxygen saturation curve, right? And you can see that at the bottom here, we do have what? We have pressure, right? We have partial pressure of oxygen, right? So we have pressure of oxygen, and then we have oxygen saturation, right? And what this means is that basically as you give it oxygen, how does the pressure change, right? Because whenever you change the pressure, it shows you whether or not we're using and transporting that oxygen, right? So this is how we measure enzyme activity when it comes to hemoglobin and myoglobin, right? This is an oxygen saturation curve. I will expect you to know about it, recognize it, and be able to reproduce it at different conditions that we'll talk about. But the first condition when it comes to this is the difference between hemoglobin and myoglobin, right? So myoglobin, let's start with let's start with hemoglobin. Hemoglobin has a beautiful sigmoidal curve, right? Has a beautiful sigmoidal curve, and this is what we see in allosteric regulation and cooperative binding, right? This is what we see. We see this type of beautiful sigmoidal curve, but then if you just have one subunit, you don't have cooperative binding, you're going to have myoglobin, right? Myoglobin, what does that mean? You see how myoglobin shoots up, the activity shoots up right away? Tells you that it's more active. It's more active. It processes oxygen faster. So myoglobin is a better transporter, right? It's a better, not transporter. That's wrong. That's wrong. It's a better holder or binder of oxygen. It binds to oxygen better, right? So myoglobin binds to oxygen better versus hemoglobin, right? So the curves are different, and then guess what? The structures are different as well, right? So this is the structure. Hemoglobin has 4 different subunits, right? It has 4 different subunits, and these different subunits are alpha 1, alpha 2, beta 1, beta 2. Obviously, alpha 1 and 2 are similar, beta 1 and 2 are similar as well. It's a heterotetramer. And then myoglobin is a monomer, right? So myoglobin versus hemoglobin, we should know about it, right? But I want you to see the red thing bound to each one of these, right? This ring is called heme. It's the heme group. What is a heme group? A heme group is what we call a porphyrin ring. Porphyrin ring. What is a porphyrin ring? It is a family of products that have this type of ring, right? So if you see a ring like this, it will be porphyrin ring. We don't just see it in heme groups, we see it in many other structures in nature. But this is the porphyrin ring, and the porphyrin ring is flat, but in the middle of it, it has these nitrogen that are obviously negative, as we already know, and then they are positioned to bind iron here, to bind iron, right? So as you can see, the one on the it's going to be on your right or your left, either way, you see this one, the unbound, right, has the NH in the middle, but then whenever it binds to iron binds here because N is negative, and then it's very stable, right? Iron binds there, right? So this is whatever you have, we call it a protoporphyrin, binds to iron, and that's what we call heme group, by the way. Heme group is whenever you have, whenever you bind to iron, then you have a complete group that's ready to bind to the protein, right? So then how does this bind? This is important, right? Okay, you have this porphyrin ring that binds to iron, no problem. We okay with that. But then how does this bind to your protein, right? How does it bind to hemoglobin? Well, look at this. This is how it looks like, right? This is what I alluded to earlier. And this is the same as this one, right? This is the same as this one. Very similar to this one, right? But you can see the heme group in the middle, right? That has the iron, right? You see the iron here nicely. But look at this. The iron is going to bind to the histidine, right? This is the histidine. If you remember, this is the side chain of the histidine. Do you remember what we call it? We call it the imidazole ring. This is the side chain of the histidine. We call this histidine F8. So this histidine binds to iron in the heme group, right? And guess what? On the other side, because this is flat, remember? This is flat. So the iron is open on the other side, right? So this side binds to the heme group, right? And then the other side binds to what? Well, oxygen, right? This is oxygen. Binds to oxygen, right? So we are binding to take histidine F1, we take the iron in the heme group, and then we have oxygen here, right? So iron is going to interact with 6 ligands, actually, even though we have histidine, which is the main one, it interacts with 6 different ligands, right? So 4 of these are going to be on the atoms of porphyrin, right? But then we're going to also have the imidazole side chain of the amino acid, right? And guess what? This one, right, the histidine F8, I can ask you, why do we call it the histidine F8? We call it histidine F8 because it is on the eighth residue in the helix, right? That's why we call it histidine, right? So whenever magnesium or hemoglobin binds to oxygen, right, the oxygen molecule is going to add the heme iron or on each ligand, right? So this is what happens. This is what happens. But let's talk more. Let's go back. Let's go back and see. Let's go back to those differences. Hold on. Fix my camera. See that it's not working. Hold on. All right. Let's go back and see more differences. So let's go back to myoglobin, right? So myoglobin is a monomeric heme protein, right? All right, so, and it's going to, the polypeptide, the myoglobin peptide is going to cradle the heme group, meaning that it just brings it in, right? And then iron, this is actually, remember if you're tracking examples, differences, I think we already talked about 3 differences between the two. This is example number 4, right? Is that iron in myoglobin is iron 2 plus, right? Iron 2 plus, ferrous iron that binds, right? But then you also have versions of myoglobin with iron 3 plus, and it's called metmyoglobin, and that one does not bind oxygen. So you definitely need iron 2 plus to bind. Actually, that's not a difference. That's not a difference because hemoglobin also needs iron 2 plus. So yeah, let's not worry about that. But in myoglobin and also in hemoglobin, by the way, right? I want you to tell me. So I did talk about like the events that happened here, right? It's like iron binds to heme group, and then the iron with the heme group binds to the histidine and other residues in this helix, right? There is a particular helix that we like, and then this is still iron 2 plus, right? And then oxygen can bind on the other side, right? These are the events, but there are more events. What happens when oxygen, right? So I think I have a nice cartoon that talks about this, right? So this helix, right? We call this the F helix, by the way. That's why the histidine is called F8, because it's the eighth residue in the helix, right? So this cartoon kind of shows you the changes, right? So when this oxygen binds, do you see like this kind of 2 things here? The heme group kind of comes in. You see how the heme group comes in like that? Comes in, and then you can see how this histidine goes in like that. And then you can see how this helix also comes in, right? So the binding of the oxygen influences starts a cascade of conformational changes, starting with the heme group. The heme group is usually flat, but when it binds, it kind of works like that, right? Back and forth, and then it pushes the histidine. The histidine kind of moves the helix, and guess what happens? When you do that, it influences the change in the entire structure, right? Now, in myoglobin, oxygen has bound, no problem. But in hemoglobin, this change in the structure propagates into the other subunits, right? Very, very important to notice. So this is a step-by-step of events that happen, right? So here, in deoxymyoglobin, right? So we were going to use this language, by the way. Deoxymyoglobin and deoxyhemoglobin is the myoglobin and hemoglobin without oxygen. So deoxymyoglobin, deoxyhemoglobin is the protein without oxygen, and then oxyhemoglobin, oxyhemoglobin and oxymyoglobin, these versions with oxygen. So we're going to use that. So in deoxymyoglobin, right, the iron lies a little bit above the heme, right? In the oxymyoglobin without oxygen, the iron lies a little bit above, right? But when you add, right, when you add oxygen, right, the heme iron is drawn in, right? It's drawn towards the plane of the ring, right? So instead of being 0.05, now it's 0.026 above the plane. See, kind of comes in. It comes in. And this, again, for myoglobin, this small change has little consequence because it's just one subunit, right? But in hemoglobin, right? In hemoglobin, it's going to make a difference because this change is going to initiate a cascade that ends up affecting the other subunits. This is the cooperative binding part, right? So again, remember, these are the events again here, right? The heme group is usually above the plane, so you have a plane. So the heme group is usually above the plane. Oh, it's actually the other way around. I just noticed. So it's usually above the plane. Oxygen binds and then brings it inside, right? So it actually pushes it this way. I was thinking the other way, but no, it pushes it towards down here. I guess it's because in the other book it's a kind of flipped so that oxygen binds to the top. But no, when the oxygen binds, this comes down, brings down this histidine, brings down the F helix, right? So be able to tell me, that's what I was talking about when I was talking about like the journey of oxygen, right? The journey of oxygen. The journey of oxygen starting from here, right? The journey of oxygen starting from it binding all the way to the types of conformational changes that it makes, right? And again, in myoglobin, this change doesn't make a big difference, but in hemoglobin, what ends up happening is this, right? These are all of the subunits, and they are kind of intertwined and commingled so that whenever a change occurs in one, propagates in the other. And it propagates it in the way that it influences the binding of the rest of the oxygen, which is pretty cool. This is cooperative binding. So that's like a big difference for those who are counting. This is like difference number, right? Number 5. Yeah. Well, we have more, right? So another big difference between myoglobin and hemoglobin is because myoglobin is an oxygen storage protein, right? You might have heard of this before, but the big differences between myoglobin and hemoglobin is that myoglobin is an oxygen storage protein, hemoglobin is an oxygen transport protein, right? Very, very big difference, right? So make sure that you remember that. Make sure that you keep that in mind, all right? So hemoglobin is not a storage protein, it's a transport protein, meaning that it binds oxygen in the lungs and then releases it in the capillaries of tissues, right? So what happens is that hemoglobin is going to be saturated with oxygen in the lung, right? And that's where I was talking about partial pressure, right? You see how partial pressure in the lung is different. So the difference in pressure influences the binding, right? So the pressure of oxygen in the lungs is going to be about 100. But then when your hemoglobin goes and gets into the tissues, it's going to be 40. And that change in pressure is what's going to release release the oxygen, right? So oxygen binding depends on pressure, right? So that's why the binding curves, right, are going to be at the bottom pressure, right? And then this will be Y, if you remember, is oxygen saturation, right? So this is oxygen binding of saturation curve. It's for pressure, right? So the oxygen binding curve of myoglobin is going to resemble the enzyme substrate saturation curve, right? It's kind of like, looks like Michaelis-Menten, right? And we do have some math that we can do about this, but I don't think we have time to do the math. We just make sure that you can recognize this curve, but then guess what? It's different for hemoglobin, right? Because you have multiple subunits, you have a nice, beautiful sigmoidal curve, right, for hemoglobin because you have cooperative binding, right? So you remember that, if you remember, when we compare them here, right? One is kind of sigmoidal, the other one is hyperbolic, right? So that is the difference between the two, right? I think we counted this difference. I was going to count it, but we already counted this difference. Now, like I said, the binding curves do not just serve for the comparison of these proteins, right? You can use it, for example, right? For here is experimentally observed oxygen curve for hemoglobin, right? And this is going to be different types of N or different types of numbers, right? And the more you add, the more sigmoidal. Basically, is what it's saying. It's kind of like, whenever you have all those four subunits, you have a beautiful sigmoidal curve. And guess what? Whenever you have one sigmoid, not one subunit, we go back to a hyperbolic, just like hemoglobin, right? So this is kind of to show you how cooperative binding is going to push us from what from hyperbolic, from a hyperbolic, is it hyperbolic hyperbole to a sigmoid curve, right? Now let's look more about the conformational change. I think we already talked about this, but oxygen binding is going to change the myoglobin formation, right? So without oxygen bound, iron is out of the heme. Like, you remember what we call this myoglobin when we have no oxygen? Deoxymyoglobin, right? So this one, whenever it's bound, it's out of plane. Whenever the oxygen binds, we get into the heme plane, right? And then whenever you go into the heme plane, you pull iron. As it pulls, it's going to pull the histidine along with it. Right? Can't see. Pulls the histidine along with it, and then the whole helix moves, right? So it pulls the histidine, and then the histidine moves the whole helix, right? So the full movement is 0.029 nanometer, right? And again, myoglobin, one subunit, no change, but with hemoglobin, big change, right? So what happens? It's going to induce a big quaternary structure. In fact, it's going to rotate. It's going to rotate the next subunit by 15 degrees. 15 degrees, right? So that's a big rotation. So if oxygen binds, so this is deoxyhemoglobin versus oxyhemoglobin. Whenever it binds, it turns one subunit like 15 degrees, and this makes a big difference, right? So again, you have a change in about 0.039 nanometers, right? So it's like you have 0.06, 0.039, right? You have a change, and this change is going to basically, it's kind of like in the same as myoglobin, but this one is going to be much more pronounced, to the point where, right, it's going to get to lead to ruptures of salt bridges, right? It's going to lead to the ruptures of the bonds, right? So this is 15-degree turns, can rupture these loosely bound. So if you remember, salt bridges are electrostatic interactions or ionic interactions between side chains. So these can be disrupted, right? So again, this is the picture that I was talking about, that leads to changes in electrostatic. So this is the electrostatic interaction. Remember, the electrostatic interaction are interactions between what? Between negative and positive amino acids, right? So that's a very important distinction, something that we definitely need to know, we need to remember. But let's look at like some of the other implications, right? So some of the physiological significance is like hemoglobin must be able to bind oxygen in the lungs, and then it must be able to release oxygen in the capillaries, right? So that is why hemoglobin needs to definitely be variable, be cooperative, have different subunits, and also be able to depend on pressure. It has to depend on pressure, because pressure in the capillaries is low, pressure in the lungs is high, right? So that is why, because otherwise, if we behaved, if hemoglobin behaved like myoglobin, very little protein, oxygen will be released in the capillaries, right? So the sigmoid cooperative, right? So the sigmoid or cooperative binding nature of hemoglobin makes it dependent on the pressure, and makes it able to be released into the capillaries, right? So another thing that we care about, and I don't want you to lose me here, okay? I want you to stay with me, right? Because now I'm going to introduce something else. I'm going to introduce the fact that in our blood, we don't just have oxygen. What else do we have in our blood, if you remember? Well, for starters, we have CO2, right? We have CO2, and CO2 also needs to be taken care of. And then also have H plus ions, right? So we have the things that regulate the pH in our blood. These, first of all, these depend, no, these affect, so they just like the amount of oxygen affect the activity of myoglobin and hemoglobin, the amount of CO2 and the amount of hydrogen also affects hemoglobin, right? And myoglobin. So I will, as I'm about to talk about, I will ask you to tell me, like, hey, if this is the normal curve of hemoglobin, what happens to it if the body has high pH, low pH, high CO2, low CO2? What happens to our hemoglobin activity and the curve, right? So don't be intimidated when you start to see some of the reactions. It's still the same idea, right? It's still the same idea, but hydrogen, right off the bat, hydrogen promotes the dissociation of oxygen from hemoglobin. So high H plus ions are going to promote this dissociation of oxygen from hemoglobin. Now, let me ask you this. What does that mean about pH? Do we have high binding at low pH or high pH? Well, low pH means high H plus ions, if you remember, right? So if you have low pH, you are acidic, you have high H plus ions, and that will promote the dissociation of oxygen from hemoglobin, right? So that is how H plus and pH affects hemoglobin, right? So make sure that you know that. We're going to talk about a few things that affect hemoglobin, and the first one is H plus ion, right? And then what else? CO2, right? CO2 also affects binding, right? So like I said, hemoglobin has a high affinity for H plus than oxyhemoglobin, right? That is why H plus promotes the dissociation, right? But so as pH decreases, right? As pH decreases, dissociation from O2 from hemoglobin is enhanced, right? So as pH decreases, as we get more acidic, we separate, right? So that is hydrogen and pH. Very, very important, right? How about CO2, right? So this, if you remember, I don't know if you remember this, but we did talk about the carbonic acid buffer, right? This is the buffer of how we regulate CO2 in the body, and it's H plus CO2 is equal to HCO3, right? H plus CO2 is equal to HCO3, and then this one will be equal to H2CO, no, not CO3, CO2. So it's like H plus CO2 is equal to HCO2 minus, and then plus H plus is going to be equal to H2CO2. 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 CO3. That's the carbonic acid. We need the carbonic acid, so the H2CO3, right? So this is, we learned about that without oxygen, without hemoglobin. But hemoglobin is going to be the H plus carrier, right? So basically what it's telling you is, this is not a full reaction. I will send you a full reaction, but this is telling you that oxyhemoglobin, you have oxyhemoglobin, right? It likes to bind to and then kind of release it. But if you were to, let's bring back the curve. As I said, this curve, we're gonna have to be familiar with this curve, but this is how pH affects, right? So you have myoglobin here, you have myoglobin, then you have hemoglobin at different pH, right? So you see how as you change the pH, as you go down the pH, become sigmoidal. As you go up to pH, we become less sigmoidal and more hyperbolic, right? So this is indicative of low versus high activity. That makes sense, right? So this is a good curve. Hopefully that's understandable. Still the same curve, but this one is showing how pH is affected, right? All right, so how about CO2? Now let me ask you this. Carbon dioxide, what do you think? Do you think CO2 increases or decreases the binding of oxygen? Right? If you were to think about what we know about CO2, CO2 will decrease. CO2 will interfere, right? So carbon dioxide is going to diminish oxygen binding, right? And by the way, this is kind of by design, and it helps us because guess what? Where do we have high amount of CO2? It's in the capillaries, right? In the tissues. This is what we want the CO2 to be gone, and we want the oxygen to be dropped there, right? So carbon dioxide is going to diminish oxygen binding so that oxygen can be dropped, right? But I can ask you, how does it do that? How does CO2 do that? This is the thing that I was talking about, right? Oh, it is CO2. Okay, the other oxygen comes from water, right? So CO2 plus H2O is equal to H plus HCO3 minus. This is part of the carbonic acid buffer, right? So CO2 and water is going to produce the H plus, and this H plus is going to promote dissociation as we saw before, right? So the CO2 is going to promote dissociation this way, right? If you remember. And as you remember, this HCO3 is the entity that's transported back through the blood and then is dropped into the lungs. So if you remember, when we get to the lung is where we're going to basically, okay, so no, I have the error here. When we get to the lung, sometimes it is transported as HCO3, but also we see H2CO3 as well. This is carbonic acid, right? Depending on what pH we have, we're going to have one version or the other, or CO2. But in the lungs is where we're going to get the CO2 version that gets dropped, and then you breathe it out. So right now, you're doing it. Like right now, as we speak, you're doing it. You are doing this reaction, and again, it's getting rid of CO2, but also this hydrogen is also promoting the this, right? And again, these protons are going to be taken up by hemoglobin, and oxygen dissociates. As we saw here, these protons, whenever you have protons, they are going to prefer binding to oxygen, to hemoglobin, and then we're going to release oxygen, right? This is how protons interfere, and they're going to do that. These proteins are going to do that, right? And as I was saying, the reverse is going to occur in the lungs, right? The reverse. So these are the reverse reactions. The reverse occurs in the lungs, in that the reverse reaction occurs, right? These hydrogens are going to bind to this to give us CO2. So if we were to look at the sigmoidal curve, right, you have the hemoglobin by itself, but then you add CO2, guess what happens? It's smaller, right? You add CO2, it's smaller. But guess what else affects it? Something called BPG, right? And I'll talk about that in a minute. But so far, we talked about, and this is another night. No, this is this is the carbonic acid that I had to write about. But honestly, I didn't have to do that, but this is kind of the summary of this. This is the oxygen, the carbonic acid buffer, right? But with this comes also how H plus, how CO2 is going to behave, right? And if you have so at the tissue capillary interface, we pick up CO2, right? We're going to pick up CO2, and then CO2 is going to go to carbonic acid, give us hydrogen, and this hydrogen is going to promote the release of oxygen because we need that, right? And then whenever we get to the lungs, it's the opposite, right? It's the opposite. The CO2 not the CO2, but the bicarbonate is going to release the hydrogen, give us the CO2 so that we can breathe it out, right? And then we're going to have low pH, not low pH, but high pH, because we're going to take up the hydrogen. So in the lungs, we're going to have lower pH than in the tissues, right? Very, very cool. But as I alluded, there is another condition, another thing that affects pH, and this is BPG. BPG is another one of those legendary molecules that everybody wants you to know about when it comes to how oxygen binding and hemoglobin is affected. But what is BPG? BPG is 2,3-bisphosphoglycerate, right? Why do we care about this? Because it is an allosteric effector of hemoglobin, right? It's an allosteric effector of hemoglobin. So the sigmoidal binding curve that we see in hemoglobin, right? We see it when it has 2,3-BPG, right? Whenever we don't have 2,3-BPG, it's actually like it's a hyperbola, right? So 2,3-BPG is a very effective, very, very effective allosteric inhibitor. Like I've seen very few examples that do that. That when something binds at a site other than the active site, it still makes this much damage. It's not really damage, it's actually good for you. But yes, so it's an allosteric effector. It looks like this, and we will learn a lot about this molecule, but it's very easy to remember, right? So glycerate, what do we mean by glycerate? It's based on glycerol, right? Glycerol, remember glycerol is 3 carbons with OH, but we call it glycerate because instead of glycerol, you have an acid here, right? If you have an acid here, it's a glycerate rather than glycerol. And guess what? Bisphosphor, because you have 2 phosphates here, right? So it's a 2,3. So phosphate on 2,3. 2,3 bisphosphoglycerate. BPG. Very easy to remember, and I definitely want you to remember it. For sure, for sure. So where does it bind? It binds in the inside, right? So it binds somewhere like in the inside of this cavity. You see how it binds somewhere in the cavity? And so it's in the central cavity, it binds inside, like right in the middle. And why is it spatial? It's because these negative charges, right? You see how like these phosphates, so it's these phosphate, as we know, phosphate has a bunch of negative charges. Bunch of negative charges on the 2 phosphates, and then you have even on your glycerate, you have a negative charge as well. And because of that, it's going to bind to all of these, right? The negative charges are going to bind to the positive charges. So it binds to 8 different positive charges, right? And then guess what? It's going to bind to different subunits as well, right? You can see how it binds to different subunits, not just one subunit. So BPG, very, very important. Very important. And as you can see, look, hemoglobin, right? Hemoglobin versus hemoglobin and BPG. Whenever you add hemoglobin, BPG, this is how it changes, right? All right, so I have one or two. I actually have two. I have two more stories. Two more stories that I want to talk to you about when it comes to hemoglobin and the things that affect it, right? I think I'm coming up to the hour, which is perfect. Right, so two more stories. The first story is fetal hemoglobin. Very, very cool in that whenever the fetus, whenever you have a fetus, they have a different version of hemoglobin, right? They have a different version of hemoglobin. So as we saw, BPG binds to hemoglobin, bends it a little bit, right? So hemoglobin usually can go up like that, but when you add BPG, it becomes more sigmoidal, right? But we have VPG, we have BPG everywhere. As we'll learn more, it's an abundant molecule, means that all your hemoglobin is always bound to BPG, right? And it's for a good reason. We don't want it to be all the way like that, because as we saw that if we don't have that sigmoidal type of binding, we won't be able to pick up and drop off blood very quickly. But for a fetal hemoglobin, you really need a higher affinity for oxygen so that we can get more oxygen from the mother, right? And because of that, fetal hemoglobin is going to have a lower affinity for 2,3-BPG and higher affinity for oxygen. So the fetal hemoglobin is a different version of hemoglobin, and we can talk about the differences and things like that, but fetal hemoglobin is going to have a higher affinity, right? A higher affinity. So because the fetus is going to depend to its mother for oxygen, right? We're going to have differences. So if we see what is happening inside, you'll see that it's going to have to replace hemoglobin with alpha chain instead of beta chain. So we're going to have different chain and different actually, let's talk about a bit about the chemistry, right? Because of these changes, we're going to not have the positive charges. And remember, BPG is very negative, it binds to the positive charges. So because of these changes and these differences in subunits, we don't have those charges. And if we don't have, it lacks 2 positive charges, to be fair, it's not all of them, right? And because of that, it doesn't bind as tight to BPG, and it binds better to oxygen, right? So this is fetal hemoglobin, and this is the curve, right? So this is normal hemoglobin versus fetal hemoglobin. You can see that fetal hemoglobin is a bit more hyperbolic rather than sigmoidal because it binds to oxygen better, right? So fetal hemoglobin, very cool. And eventually we stop making it later. But the last one that I want to talk about is sickle cell anemia. Sickle cell anemia is a single mutation in hemoglobin. Single mutation. It's actually crazy, right? You're going to have where in the beta chains, right, glutamine is going to be replaced with valine, right? And as we've learned in protein structure, we know that this is a destructive mutation, right? We had glutamate, not glutamine, glutamate, right? Which is big and positive, versus valine, which is small and hydrophobic, right? So it's a big change in this beta chain. And whenever you see that, it's going to cause a change, right? So this is going to cause a big change. This is kind of a cartoon, but obviously it's not going to be like that. But it's going to it's showing you that in sickle cell you have a different type of the way they're going to get together and kind of stack together, and the way hemoglobin stacks together is important, right? So usually the hemoglobins are not going to stack together, but here they create these types of grooves so that they can clump together like that, right? So in sickle cell anemia, the hemoglobin is going to clump and aggregate together rather than be free. And because of that, because we know that hemoglobin is one of, is basically, these red blood cells are mainly made of hemoglobin. So if hemoglobins clump together, these cells are not going to have this nice disc shape. They're going to have a crescent or moon shape. That's why we call them sickle cell anemia. But also another big important thing is that if these hemoglobin proteins are kind of clumped together, they're not going to transport oxygen efficiently, right? And this is a devastating disease. A lot of people don't know this, but over 90% of people who are born with it do not live to be even 5 years old, right? Most people die in their infancy, and the people who survive, they live with excruciating pain. It's kind of messed up, but again, this is kind of the structure of how the clumped oxygen, the clumped hemoglobin is going to be. Something actually really cool is I made a whole video about it, so you can find it on YouTube. But CRISPR, the new gene editing technique, has just cured sickle cell anemia. Cured. But they did cure it. The people who were cured have not had a recurrence. The only problem is that the treatment was $2 million, so it's not really ready to be scaled. But the way they were cured, it's crazy. They took advantage of the fetal hemoglobin, actually, right? So they found a way to genetically engineer the genes to produce fetal hemoglobin again. And by producing fetal hemoglobin again, the red blood cells are now using that hemoglobin that is even better than the normal one, and they go back to normal. It's crazy. It's incredible. And it works. It's just very expensive because they have to take out the patient's blood and change it and then bring it back and do it enough so that all of the cells, at least the blood cells, will be replaced. But very, very exciting. That's now that you know about fetal hemoglobin and sickle cell anemia, you can actually explain what's happening, which is really cool, right? Another thing that I will talk about, last thing this time, I promise, is that another molecule that binds to hemoglobin is nitric oxide, right? Nitric oxide has a pretty high affinity for hemoglobin, right? It binds much more tightly than oxygen, right? And it can be good, and some people have been thinking about using this as a, okay, you should see it here. Some people have been thinking about it and using it as a therapeutic agent, but also it can be used, it can cause problems, right? If it's competing with your oxygen, and it competes much better it can be used. But we you just know about that because there's so much that goes into it, to the point where some people are starting to just take nitric oxide based pills and stuff like that. But this is another story. Just know that nitric oxide is another molecule that binds to protein, binds very, very highly. But I think that is enough. I think that is enough. That is a good place to stop. And hopefully, this video was enough and I was able to convey all the information that you need to know. I'll try to cover all the bases and make sure that you understand the importance of myoglobin, hemoglobin, and their differences. Make sure that you know 5 to 10 differences between them, and also the journey of oxygen and what it does and what's happening to the proteins, from the chemistry and the binding and conformational changes and the transports, and then end with the things that affect this activity and the transport. So we talked about pH, CO2, BPG, nitric oxide, or what happens when you're sick, when you have sickle cell anemia, or what happens when you are a fetus, which is fetal hemoglobin. So I think that's enough. But if you have any questions, you know how to reach me. I will see you in the next lesson. Bye bye.